It is my very great honor to introduce Andrea Tilden. Andrea is the J. Warren Merrill Associate Professor of Biology. She earned her PhD in zoology at the University of Oklahoma. A neuroscientist, she studies the influence of the environment on cellular and molecular neural function in collaboration with many of her students. She founded the neuroscience program here at Colby. She was the 2009 recipient of the Charles Bassett Teaching Award. She's the co-director of the Colby Achievement Program in the Sciences, and she is also the co-director of our NIH Embrey program. Welcome, Andrea Tilden.
Thank you, Provost Kletzer. It's a great honor to be here on this wonderful occasion. Everybody, there's a lot of enthusiasm generally campus-wide, and so this is a very exciting time, and I'm honored to be a part of this. The question why we sleep seems like an obvious one. There are many reasons that we sleep. We sleep because we are tired, but I'm interested in that question at a deeper level. We know some of the things that happen when we are sleep deprived. So, for example, irritability, cognitive impairment, memory lapses and loss, and even longer-term, more serious diseases such as type 2 diabetes and other things may be a result of chronic sleep deprivation. And by the way, we are chronically sleep deprived. And if you look back over human history, you'd realize that we probably evolved to receive more like 10 to 12 hours of sleep rather than the 4 to 7 hours of sleep that we may be getting now. I'm interested in a hormone that's involved in the sleep process, and this hormone is called melatonin, and some of you may have heard of it, some of you may have taken it. You can go to any Rite Aid pharmacy or even convenience store and buy it, and you can even go to the drink section and pull out something, I think it's called Bob Marley's drink, and it's got melatonin in it. This is not good, and I'll tell you why this is not good. Melatonin is a hormone. So melatonin is a hormone that's produced in your brains in response to environmental darkness. We produce it when we're sleeping. And what happens here is, here's the pineal gland, the gland that's responsible for making melatonin. The pineal gland is activated to make melatonin when it's dark out. So your eyes are the sensors that receive that light and dark information and send it through this convoluted pathway. It's even more convoluted than this, back up to the pineal gland where it starts making melatonin when it's dark. The melatonin then goes into your bloodstream and travels to all of the cells and tissues in your body to tell you that it's nighttime. Therefore, we call it the chemical messenger of darkness. I love that term. And it's got lots and lots of physiological functions. So it's the signal to your cells that, hey, it's nighttime. And what it does in your cells is it has to activate or inhibit a number of different processes that your cells need to undergo when you're sleeping. Another function of melatonin is that it's a free radical scavenger. It's an antioxidant, in other words. And many of you take vitamins that are antioxidants, and you may have heard of free radicals causing damage and disease, cancers, age-related diseases, et cetera. So that's a big area of research right now also, is this idea that there are molecules that can scavenge up your free radicals and potentially protect against diseases. And that is a part of the area of my study. But many of the functions of melatonin are still largely unknown. And I should point out right now that I've been studying melatonin for about 30 years. And everything that we know about melatonin tends to lead, like any question, a good question in biology, tends to lead to more questions than it answers. So I'm going to focus this talk. There are several different areas of research, but I'm going to pick one in particular. And I'm going to talk about what may be melatonin's role in sleep with regard to our brain cells. So I said before that sleep deprivation leads to memory loss, irritability, learning impairment, and other problems, things that students should take note of, because when you don't sleep, when you are sleep deprived, then these functions all decrease substantially, and tests have proven it. Psychological tests have shown this. I work, again, more at the molecular and cellular level, so I'm going to focus more in that area, but I will bring it back around to what the implications are for memory. A lot of memory-related processes occur in this region of the brain called the hippocampus. This is a paired structure on the right and left hemispheres of the brain. And there are cells in here that are involved in creating new memories. And in fact, if you have damage in this area, you are unable to make new memories, and it leads to all sorts of problems with creating and learning new things. Amnesia, basically. So the hypothesis in my lab is that melatonin, because it's released at night and it goes everywhere in the body and can have wide-ranging function, may have some function in the brain. And so one of those functions may be in increasing brain cell growth. Now, there are two ways a brain cell can grow. One is to divide and become more than one brain cell, and another is to grow connections to other brain cells. The connections to other brain cells part is by far the more prominent thing that the brain does to make new memories and to learn. So by connecting to new brain cells, the brain, in ways that we are only at the very beginning of understanding, will create new memories. Another hypothesis is that melatonin also may scavenge free radicals to protect against oxidative stress and free radical damage, and maybe that's happening at night as well when your melatonin levels are high. We operate in biology on a principle called the croke principle, and I talk about this extensively in my comparative animal physiology course. And this is the principle that there's the ideal animal to study any biological problem. The ideal animal for my studies is crustaceans, and they come in a variety of forms, and I study a variety of different crustaceans. And the animal tends to suit the question. So if I'm studying larger scale processes using organs and organelles, I'll study something like a lobster. For smaller intermediate questions, particularly behavioral questions, we do a lot of work. When I say I really mean we, because as I'll say later, my students do the vast majority of the physical hands-on labor. But we do a lot of field work where we're looking at animals in their environment, and this is an invasive species, green shore crabs in Maine. They've been around for a while now, and they are decimating the flora and fauna of the coastline, and so we have an interest in them as well. Sometimes these smaller fiddler crabs, and then sometimes we even study these guys, which Whitney probably knows well, and we call them in my lab pond scum. But these are actually little animals, and they are almost microscopic. They're about 1 millimeter, and if you take a jar of pond water, they're the little tiny white flecks that appear to be jumping up and down in the water column. They're useful not because they're small, but because they're the only crustacean whose entire genome has been sequenced right now. So lots of animals now, including us, have had our genome sequenced, and this is my model because it's got a fully sequenced genome. This is a video showing you what it looks like when we study the brain cells. So I'm going to talk first about the brain cells, and then I'll talk about the molecules. So what we're studying in the lab is the way that these cells here, this is a round cell body. When we take brain cells out of a crustacean and put them in a culture dish and add nutrients and other supplements, they look just like this round body with none of these projections. These projections are the connections, and we call them neurites. They're axons and dendrites, and you may know them commonly as that. They're the ends that release neurotransmitters like serotonin and other things. So we're interested in the growth of these neurites. And when we first put the cells in culture, these neurites are so delicate, they have already fallen off by the time we get the cells there. So we're starting off with a blank slate, and we can watch the growth of the neurites. And it's really fun and fascinating to do, so much so that I'm going to show you. I'm hoping that this video will go. Maybe not. Doesn't seem to want to. Okay, here it goes. Okay, so just watch these projections over time. And so what's happening is this cell is investing a lot of energy into creating these new connections, and here's this one lonely cell in this culture dish. There are some other cells around just outside of view and sending out chemical signals to tell this cell to make a connection to it. So we observe the growth. This is a time-lapse image, so it doesn't really happen this quickly in real time, but this is over the course of about a day and a half. The stuff going by is just debris. And over the course of 24 hours or so, you get very extensive growth of these neurites. And what we do in the lab is we quantify this. And so we use computer image processing and other sorts of programs. We take images of the cell at various stages, and we quantify the amount of this growth. And so here's just a still cell, but shown a little more close-up and in a little more detail. So this is a cell grown by one of my students. We grow thousands and thousands of these cells and monitor thousands of them. They're really beautiful. They just start out as the round cell, and then this is a 24-hour picture showing the growth. That's the cellular side. And the upshot of the cellular studies, I'll just go back for a second. The punchline for what we've studied so far is that when we treat these cells with melatonin, indeed, these neurites grow, and they grow at about twice the rate that they grow when the cells are untreated. And this suggests to us that maybe what melatonin is doing at night in these hippocampus-like cells in the crustacean, and perhaps in us as well, is help to solidify memories that we've had during the day. And I'm not the first one to come up with this hypothesis. This hypothesis has floated around for a pretty long time. But in our lab, we've hypothesized that melatonin may play a role in this. And so this is novel. Our next question is then, we know that melatonin is enhancing these neural connections, so how is it doing this inside the cell? So we looked at outside the cell with the neurite growth, and now we can go into the cell, and this diagram should be intuitively obvious what's going on here, which is, this looks worse than Whitney's, isn't it? But what I want to point out here is, it is complicated, and that's why I put it up here. There's years and years of projects here for my students and me to study, because melatonin may be affecting any of these things. These red arrows here each represent different genes that may or may not be turned on by melatonin or may be turned off by melatonin. All of these circles are proteins that may in some way interact with melatonin, and all of these are involved in day-night activities of the cell. And you have these in all of your cells, and they're called molecular clocks, and they're ticking away. And so that's what we're studying now, is the molecular basis. And we're at the very beginning of this study. I do have data, but I haven't even analyzed the data in this part of the study yet. Research is a very collaborative endeavor, and I work a lot with students, I work a lot with colleagues. One of the places where I do a lot of my research, and have been for about the last 20 years, is the Mount Desert Island Biological Lab. I've been at Colby for 16 years. Before that, 15 years. Before that, I was at Macalester College for 5 years. I'm from the coast of Maine, just a few miles from this lab. And for me, at Macalester, this was a way for me to get back to Maine, be near my family in the summer, and be able to work with marine animals that I was interested in. So I started this collaboration a long time ago. And then when the job ad for Colby came up, I just jumped on it and said, this is perfect for me in about 27 different ways. And I've never looked back. I've been just so happy here ever since. And I'm only about 2 hours away from my other lab. So we do some of the work in my lab here. The most of it, time-wise, occurs here. And it occurs during the summer and during the academic year when students are doing independent studies. And then in JanPlan, and sometimes in the summer, my students and I will go to this lab. This lab started out, when I first started going there, as a series of little shacks right on the ocean. And they had running water and electricity and not much else. And it was a great place for the meeting of many minds from lots of different institutions. It was great. But since then, in the last 10 years, they've grown and flourished, and now they have this Catherine Davis Center for Regenerative Biology and Medicine, the same Davis family, by the way, that gave us the new science building here. Research like ours takes money, and it's very expensive. The animals themselves, we can dip them out of the pond and collect pond scum. We can go on the coast and collect them, and we do. But all of the other machinery, the instrumentation, the microscopes, the chemicals are very expensive. It takes a lot of money to do this kind of research. And we get a lot of our funding for biomedical research at Colby through the National Institutes of Health. And in particular, we have this grant called the NIH Embrey Grant for Maine. It's a statewide grant. And this is a grant that started about 10 years ago by Patricia Hand, who's in the center here, who basically rebuilt that lab and got those new buildings built and brought in this funding to Maine that is spread far and wide. As you can see, there are lots of institutions involved, including Colby, Bates, and Bowdoin. This is my colleague, Patsy Dickinson from Bowdoin. She's a neuroscientist there, and we collaborate a lot as well. So lots of collaborations have been enabled by this grant. And two of my colleagues, Judy Stone and Lynn Hannum, and I, the three of us have been steering this grant for the last 10 years and now another 5 years into the future. And it's also interesting to note that at least 5 or 6 million of that over the 15 years comes directly to Colby for paying for student summer research assistantships and instrumentation, microscopes, molecular probes, other sorts of things, and research technicians to run our machinery. The most important part of my research is the students themselves. I've been working with students forever. When I first got to college, I went to a small liberal arts college, and I said, oh my God, this is like the perfect job. I'm going to be an art professor. And I didn't. I ended up taking a biology class, and it wasn't the biology, I have to confess. It was the really fantastic professor who just wowed me and made biology interesting. And that's why I'm a biologist. It has nothing to do with a deep, long, enduring love of biology. I acquired it. And I worked with professors in college doing similar sorts of things, and I've always enjoyed working with students. I have Colby students who work with me during the academic year, during the summer, and also during a Jan plan. And as I mentioned before, I have a Jan plan where we take students for 2 weeks to the Mount Desert Island Biological Lab where we intensively work on the molecular work. This is my research lab here. This is Emily Berner, who's working with me, and Patsy Dickinson from Bowdoin. But this is her in that Jan plan. And then here we are at the Mount Desert Island Lab here and here doing molecular work. Research is very intense. It's a very social endeavor as well. Students come up with lots of great ideas, and they're highly engaged. And these are students who are giving up everything else in Jan plan. I'm talking about skiing, partying to some extent, all sorts of things, to spend a ridiculous amount of time in my lab because I'm a circadian rhythm biologist, which means I study things that happen all throughout the clock. And sometimes they spend 24 hours in the lab sampling certain things, and they really work intensely. There is always a lot of thumb wrestling that occurs in lab work. This is probably, we often have 1 a.m. or 3 a.m. lab meetings when everybody just pulls together and says, okay, here's what we've got to do because we've got to get a certain amount done in a rapid time. And so Colby students always have amazed me at their engagement in this process and willingness to do this. And year after year, I can get at least 10 students willing to give up everything and spend Jan plan with me doing this. I have summer students as well. Here we are. I'd like to say we're out here sampling, but really we were out at Margaret McFadden's place going kayaking because it was a really hot day. But this is what it would look like if we were out sampling stations out in the pond. We are not sampling here either. This is us at Acadia during Jan plan, experiencing life there. And at our lab at Mount Desert Island, my students like to take pictures at various times of year of the different sunsets. Here's a winter sunset and a summer sunset, which is sort of a nice finale to talking about sleep and circadian rhythms. Seems to like to blank out on me. The last slide was just showing a couple of publications that have come out of our work. These are some of my more recent publications just in the past 5 years or so. And in blue are student co-authors. And students have always been an integral part of my research at Colby. It's the reason that I do research, really, because otherwise I would not be interested in doing research in an environment in which I was working by myself. So I will end with that and take any questions.
Professor Tilden is too modest about her role in these National Institutes of Health grants, but I won't let that stand in the way of questions. So the floor is open for questions. When we get you a mic and you ask a question, would you stand, please?
Hi, Andrea. Are there any functional differences in memory for people who are blind?
Oh, that's a really interesting question. And I don't know that that has been studied, but I will, because you bring up blindness, I will bring up one thing. People who are blind and who have, it is often the case, at least in the past, that people would have their eyes removed if they were blind, because eyes can acquire a lot of damage just by opening and closing without you realizing what's going on. I mean, we blink reflexively because we can see. So people who are blind and have any sort of nerve damage in the optic nerve do not produce melatonin appropriately and have all sorts of disruptions in their circadian rhythms. Yeah, so that's a really good question. And so it follows that naturally, if they're not producing melatonin at night, they might have problems with memory. And like I said, this connection with melatonin and memory is at its very beginning phases. We did our study that came out in 2012. Since then, they've built upon that and done mice studies. Not me, but people who study mice have looked at it in a larger scale, actually looking at memory formation, because you can do a lot more with memory in mice than you can with crustaceans, because they just don't do much of anything. Mice, you can follow their behavior, you can relate to them, you can know if they're remembering something. Not so much with crustaceans, but that's a really good question and a good area of study.
Okay, we have a question there, and then if we can follow up with a question in the front.
Yes, I'm looking for news I can use.
Steve, can you stand? Would
You
stand,
seemed a little
please?
skeptical about melatonin sold in corner stores, and yet you talk about having students up at 1 a.m. and 3 a.m. in the morning. And so I'm wondering, are there any inside tips that you use to keep them going like that?
I know, I'm sort of a jerk, right? Yes, I do have tips. So melatonin is produced at night in response to darkness. It's on a rhythm, meaning that that rhythm will continue to persist unless you do something to mess it up. It's genetically driven, but it's also kind of fine-tuned by melatonin. So my advice, if you're going to pull an all-nighter, is to take the melatonin, even if you're not going to sleep. The melatonin, if it's there doing some good, it will be doing that good even though you're not sleeping to produce it. The stuff that you buy at Rite Aid or in the Bob Marley drink is identical to your own hormone. So I can't stress that enough. You are actually consuming a hormone. And furthermore, there's probably nothing wrong with it. People have been taking it for about the last 25 years anyway. And it doesn't seem to cause any substantial damage that anybody knows about in the short term, in 25 years. But if you're going to take it wisely. In other words, take it when it's likely to naturally be produced to supplement when you would normally take it. So, for example, take it in the evening just before going to bed. Don't take it in the afternoon. Don't drink that Marley drink in the afternoon, because it will completely screw up your circadian clock. That's why I was not very excited about the drink.
Had that been your question? Okay. Another question. Oh, I'm sorry, couldn't see that.
Andrea, I'm interested in whether any of the work that you've been doing on melatonin has implications for, I'm particularly interested in the neuroprotective effects, Lou Gehrig's disease.
Yeah, that's an excellent question. So melatonin has been studied in relation to all of the different neurodegenerative diseases, and it's a flourishing area right now of study. And so yes, Lou Gehrig's disease is a prime target for a potential therapeutic value of melatonin, as well as Parkinson's, Alzheimer's. There's a breast cancer connection. There is a reproduction connection. There are so many connections because sleep is so important, and because melatonin is an important signal, as you can imagine, is probably involved in lots of these different issues. And is it a cure-all for some of these things? Probably not, but it could be a piece of a bigger treatment puzzle. And they're already treating certain breast cancers and certain neurodegenerative diseases with melatonin supplemented with other known treatments, and it seems to enhance those treatments. So lots of potential therapeutic value.
Okay, Andrea, thank you very much.